Swayam Prabha. Digital India. Educated India.
So how can we generate in the laboratory diversification that is at the end the solution to this optimization problem? Well, some of the tricks that we use in the laboratory to this end have been mentioned in previous classes. One of them, that to me is a very, very powerful one, is this targeted evolution of genes. I mean, when I put Pseudomonas, forget Pseudomonas, you can put any other biological system, any other bacterium. So if you remember, and if not, I refresh your memory, there's this technology called single-stranded DNA recombination that is about entering in the cells mutagenic oligonucleotides that penetrate, that invade the replication fork, and then as inherited as part of the replication, say, process. So the kind of essence of this process that cells grow and then the DNA replicates, and in the moment that the DNA separates for replication, then we can invade the replication fork with a complex between an artificially produced oligo that is mutagenic and these proteins that are able to stabilize the oligos and targeting them to the replication fork. Okay, well, this trick has been used repeatedly in my laboratory and many other laboratories to bring about diversification of a specific genomic segments. Okay, so imagine that you have a given DNA, and you know the DNA may be various kilobases, but you want just to hyper diversify just one little segment of that DNA without diversifying the rest. Okay. So what you can do, and I see that we have done very, very, very many times, is to develop an oligo to synthesize artificially in chemical synthesis, an oligo that carries a mutation or a library of mutations that you want to insert in that particular location, and then in trans you may produce a protein. In this case, it's a protein called SSR, but there are many others that can be expressed through a device that responds to benzoate. And then when that happens, then the protein will be associated to the oligo and will penetrate, will invade the replication fork, and therefore mutagenesis will happen. So for instance, one of the ways of optimizing some intergenic regions with this technology is by having pools of oligos that are not, where you have some positions in the oligo that are not defined, so you have a mixture of them. Then you enter that pool of oligos in just a precise site of the genome that you want to hyper diversify, and then you have a readout, for instance, in this case GFP, then you can select visually those that will do the job better and that are optimized for a given function. Okay? So in this case, for instance, it was an experiment in which we try to optimize the Schneider-Gartner sequences, the ribosomal binding sites of a promoter that was in front of a GFP protein in front of in the genome of Pseudomonas putea. So what we did was to transform the strain with a pool of mutagenic oligonucleotides that would target specifically that region, and at the same time express in trans a protein called SSR that will bind the single-stranded DNA and would take it to the replication fork. And then by doing that, you can generate a great diversity in that particular place, and obviously as we were looking for an optimal or a very, very strong ribosomal binding site, then the system gave us the solutions that are at the top there. Okay, so this is something that you can do. There's another, in my opinion, super smart approach to hyper diversify specific chromosomal segments, sparing the rest. And in this particular case, it goes as follows. If you recall, well, you know, sometimes I talk about things and I assume that you remember what I said in previous classes. I'm not sure about that, but in any case, I'm mentioning here two things that we have discussed in previous classes. So on the one hand, we have a T7 RNA polymerase that, you know, is able to recognize, and a specific tRNA promoter is an orthogonal system for transcription, because T7 RNA comes from a phage and therefore is orthogonal. But at the same time, a part of being a polymerase, what it makes is that it jumps into DNA and then it moves along the DNA until it finds a terminator. Okay? So someone had this bright idea. What if we limit a chromosomal segment between a T7 promoter and a T7 terminator? And let's imagine that I want to hyper-diversify. In between, I want to spare the rest, and I want to hyper-diversify just between here and here. Okay, so to do that, what you can do is to make a hybrid protein between this T7 RNA polymerase and a cytosine deaminase. Cytosine deaminase are these base editors that we discussed yesterday, and what they do is, yesterday or the day after, before yesterday, I don't remember, that the moment they find in the DNA a C will be converted eventually into a T, okay? Okay, so the idea then is to say, well, if you express in vivo this hybrid between T7 RNA polymerase and cytosine deaminase, the prediction is that the complex will jump on the T7 polymerase and will proceed transcribing, but in that case transcription is not of interest for us. The only thing that matters is that the RNA polymerase will pull the cDNA, the CDA, the cytosine deaminase, through the entire DNA segment in such a way that they will read the whole sequence, and every time or with a given frequency that this finds a C here, bingo, it will be converted into a T here, eventually, okay? Okay, so what is the consequence of that? That after this process happens, then this gene here will be peppered with mutations with a given frequency in the positions that are cis there, okay? So, and there's a super good procedure for hyperdiversification of given chromosomal sequences. So imagine that you have a situation where you want to keep in good shape a whole genome, but there's one gene that you want to hyperdiversify because you think that by hyperdiversification of that particular gene, then something happened that is of biotechnological interest. Well, this trick is optimal to that. Just to show some results, you don't have to go into the details, but this shows, for instance, you have this PIR-F gene. So this PIR-F is in the chromosome of Pseudomonas putida, and it creates sensitivity to, well, it's a gene that is involved in uracil metabolism, but it's also a gene that makes cells to be sensitive to an antibiotic that at some point we didn't want it to be sensitive to. So what we did as a kind of proof of concept was to take this gene, flank this gene by a T7 polymerase, an T7 promoter, and then apply this kind of rowing, say, passage of T7 polymerase hybridized or linked to a cytosine deaminase, this base editor, and then what we obtain is a segment of DNA that has many mutations. Okay? So that's something that is very nice, and you have a selection system, then it's wonderful because you can generate diversification very easy. Okay, so in sum, to solve some problems of optimality, we cannot use first principles, we can use evolutionary approaches, and the evolutionary approaches can be more general, where you just let the system to fluctuate on the basis of stress, on the basis of ROS. You can go more concentrated into given locations of the chromosome. So what could be some applications of this, that you can really visualize the power of the technology? Okay, so I'm going to show you some instances that are connected to things that I also mentioned in some of the earlier classes that are about generating adhesions that bring about contacts between cells and a given surface, or bring about contacts between different cells, different types of cells, to create synthetic consortia. Okay? So now at the end, you may remember that the trick is having an antibody that will recognize a given antigen. So how do you get antibodies? Well, the standard way to get antibodies is to inoculate an animal and then expect that the animal will be able to produce an antibody of interest. But you can make this in a completely synthetic fashion using the approaches that I just mentioned before. How can you do that? Well, if you want to have an antibody that recognizes a given antigen, then what we do is that you put this antibody, the gene, you codify, you encode this, the sequence of the antibody fused to a display system in the chromosome of a bacterium, of E. coli, and then by doing that, then you expose on the surface the antibody, or at least the business part of the antibody, the part that recognizes the antigen. You put it on the surface in such a way that now the cells are coated by this antibody. Okay? But as the gene of the antibody is encoded in the genome, then you can make the trick that I just mentioned. You can make this hyperdiversification of the so-called CDRs, that are the sequences of the antibody that recognize the antigen. So if you now hyperdiversify at the genomic level this region, this region, and this region, what you do is that you generate an immense collection of antibody variants that will go into the surface of the bacterium and that you can select by different means. So for instance, in this case, you can select them through immunomagnetic beds. Okay? So that's wonderful, because at the end what you are making is to have a kind of entirely synthetic immune system. So to get antibodies, then you need a camel, or you need a bacteria, or you need rabbits, or you need mice, or whatever. In this case, it's all in vitro, and you generate antibodies that are as powerful as the antibodies that you get inoculating an animal. Okay? So this is great, not only for, say, the well-being of the animals, but also because this is a very, very quick system to develop antibodies against new targets. Just as a way of anecdote, when we had the COVID epidemics in Spain and all over the world, we were able to very, very quickly develop antibodies against some of the variants of the virus, of this famous protein that is the spike protein, because we could have this spike protein produced in vitro, and then through this immunomagnetic trick, we could isolate very, very quickly variants of antibody that were really, really neutralizing, at least in vitro, and that could be isolated very rapidly in a question of days rather than in a question of months, as is the case when you have to inoculate an animal. Okay? So I think that that's something that it was one of the cases where you notice that solving the problem of creating a good antibody against an antigen, that is a super complex endeavor. If you do it through first principles, then you can also solve the same problem by making this combinatorial approach followed by a good selection system. And then when you have these antibodies on the surface and everything, then you can do the type of things that we have been discussing in previous classes of making clusters or making bacteria stick into surfaces and all the rest of it. In fact, we had one of the presentation of the papers had to do with that, and just as an illustration, you may have seen both in my presentation and in the presentation of one of your colleagues this type of graphics here. But what to me is more kind of visual is that the bacteria that are coated with these antibodies really get stuck to the surfaces, right? To the point that the shape of the bacteria gets deformed and they really get stuck to the surface because of the action of these antibodies that are on the surface of the bacteria and are binding the target antigen that is in the surface here. Okay, so that's something that is quite nice. Okay, so another way of optimization. So in this other case, what we tried to do was to optimize an optogenetic device. What is an optogenetic device? You may remember, again from the very first lecture, is one system that will react to light by activating transcription of a target promoter. Okay? And if you remember this photograph, this E. coli photograph that I showed you the first day, then I didn't give you details, but the system is based in a complex between this transcriptional, this sensor that is called CCCAS-S that recognizes a small molecule called PCB. PCB is a chromophore. Is a light-sensitive molecule, and then there is this two-component system, and then a transcription is initiated. Okay, so we wanted to optimize this system. Was initially device for working in E. coli, and it's the same system that you saw in the photograph of that guy that I showed you the first day, and we wanted to optimize it for Pseudomonas, because when you move from one place to the other, the parameters change. So what we did was to apply the same principle that I showed you before with the antibodies, but in this case, applying that to a whole, say, pathway. This is not a metabolic pathway, it's a device, it's an optogenetic device that is intended to bring about expression of genes of interest when cells are illuminated with green light. Okay, so we have a bunch of parts here because we need 1, 2, 3, 4 promoters. We need 1, 2, 3, 4, 5 ribosomal binding sites, and we need 1, 2, 3, and 4 terminators. Okay, so we have a bunch of parts. So if we had to optimize this device on the basis of the parameters that are available by the different parts, it would be impossible because there are so many parameters that it will be very, very difficult. Okay, so what we did was to target the regulatory regions, the promoter, the different terminate, the different ribosomal binding sites and so on. And at the end, what we obtained was a library, a combinatorial library of many, many different variants of the same, say, basic structure. And then what we did was to isolate specific colonies with a colony picker, and then to go one by one, and then to measure light emission in response to fluorescence emission on the basis of light exposure in a cytometer, in a fluorescence cytometer. And then eventually, with this type of setups, then we achieve some bacteria, some clones that responded very well to light and did not respond at all when the cells or the cultures were not illuminated. Okay, so these are some of the, I mean, this is like the starting point, and this is the final point. Okay, so well, you can go from here. You make first a selection, and then you have a kind of number of cells that look promising. When cells are illuminated, this is just a microtiter plate that is illuminated or not illuminated with blue light. And this is the green light, and then this is the result. Okay, so at the beginning, all of them are responsive to light, but many of them have a high basal level of expression if they are not illuminated. But after various rounds of mutagenesis, then you get this library or this specific set of clones where you can really see that in the darkness they don't express any gene, and in the light they express genes, the gene of interest. In this case is a proxy, in this case is just what I said, GFP, okay? And this is, visually is very nice. So in the darkness you see no emission, and when you illuminate the cells, you see that, you know, GFP gets expressed at high levels. So this is really nice, and you can see the difference between non-illuminated and illuminated, and they are very well regulated. Okay, so this is a case of typical, say, optimization of a device where you have many, many different parts at stake, and you don't know how to make them work from a kind of completely rational perspective. Okay. Okay, but the story does not end here. Okay, because we can generate diversification not only by playing with the DNA, but with playing with other things. Yeah? And to give you an introduction of what could be these other things, then we need to move a bit to another story that is about how cells are organized inside. Yeah? And as you will see later, the connection between how cells are organized inside and how that can be leveraged for producing another level of hyperdiversification, and therefore a better and more granular exploration of a solution space for a given problem. Okay. So when one thinks on a product, on something that happens in the cells, it can be a bacterial catalysis or some other process, then this is the kind of naive view. Okay. So you have an input, you have an output, and something happens there. Okay. And what happens in many cases, we just visualize it in these terms. we have some genes, we have some products, we have some metabolic connections and everything. But what we sometimes forget, or we prefer not to be aware, is that the interior of a cell is an authentic factory. It's a chemical factory in miniature. And for a cellular cellular factory to work, you need to have a degree of three-dimensional architecture. You need to have pipes, you need to have valves, you need to have channels, you need to have different, say, physical, say, aspects that make the system to work as it does without mixing everything with everything, because then the cells will not work at all, okay? So when we take a closer look to the internal organization of cells, what we observe is this kind of messy or apparently messy mixture of DNA, ribosomes, different proteins, and so on. So it is logically impossible that this system works unless it had an internal architecture that enables it to work, okay? So there is this view that in fact cells have a layer of regulation and a layer of functioning that is not related to its biochemistry, it's not related to their genetics, it's just based in the physical structure that you have inside the cells. And therefore, by the same token, that in a factory you have valves, you have channels, you have different, say, physical structures, then it is likely that cells have a layer of control that is like if it were a kind of super small nanofluidic system, right? In such a way that inputs and outputs are processed not only through a biochemical level and through a genetic level, but also through a kind of purely physical level, okay? And therefore, that takes us to the problem of the organization of the hardware of the gene expression flow. If you think about it, up to this point, the only aspect of the functioning of a cell that you have considered in synthetic biology has been the software. Software here, software there, DNA here, DNA there. But how about the hardware? So if we have a plasmid in a cell, where is the plasmid? Where is RNA polymerase? Where are the ribosomes? Where are the promoters? Where are the transcripts? So where are physically, not just as a sequence, where are they physically? Okay, so in the last few years, there has been a tremendous advance on methods for visualization of where things are inside a cell. And just as a way of example, we have been working for a long time with a plasmid that is called PWW0. The role of it I will not discuss. But one of the ways of visualizing where the plasmid is, and as you can see, the plasmid is not all over the place, it's just in a specific spot of the cell, is by entering into the structure of the plasmid an operator sequence that can be bound by a repressor that is itself joined to a fluorescent protein. Okay? So that means that you have this target site in the plasmid, and then you produce from somewhere else a protein that recognizes this binding site there. You can look at the microscope, and as one of the partners of the protein hybrid give you fluorescence, you can immediately identify where it is. Okay, so, but you can make more questions, not only where the plasmids are, where is RNA polymerase? So one way of looking at that, and there has been tremendous advances of that, is that you can make a fusion between one of the genes that encode one major subunit of RNA polymerase, that is called RPOC, and then you make a fusion with a fluorescent marker, for instance, with monomeric superfolder GFP. And then you look at the cells, and then what you obtain is this again, that the polymerase is not all over the place, is found in specific, say, sectors of the cells. And interestingly, if you stain the cells with a dye that is called DAPI, then the DAPI binds the DNA. So we have like two fluorescent markers. One of them reveals the position of RNA polymerase, the other reveals the position of DNA. And what you obtain when you look at the cells is a perfect overlap between the signal that comes from from DNA, and the signal that comes from RNA polymerase. Okay? So it seems then that what is called the nucleoid, that is where the DNA is inside the cell, overlaps 100% with the location of RNA polymerase, and that in principle may make sense. One thing that is interesting to consider when you analyze these images is that in an image you have a bidimensional image, right? But in reality, cells are three-dimensional. So you have to be careful on how you interpret the signals, because if you have an homogeneous distribution of a signal in a fluorescence microscopy, then what happens is that the signal that comes from the center of the object is more intense than the one that comes from the periphery, from the margins. But if you have an object and the object is, and the fluorescence comes from the boundary of the objects, then what you observe, or you should observe, is that the signal at the sides should be more intense than the one you have at the center, okay? So these are things that microscopists know very well and that allow them to really do interesting things. Okay, so when you compare where is RNA polymerase versus where is DNA, you have a perfect overlap, okay? And you can, with the techniques available up to this point, you can get images and to make one by one a good analysis of where the different signals are and so on. And what you obtain when you measure many, many cells is that there's a perfect linear correlation between the location of RNA polymerase and the location of DNA. So far, so good. But where are the ribosomes? Okay, so we can use the same technology to take a look to the ribosomes, and then what you observe is that the ribosomes are distributed also in specific locations. They are not all over the place, and you can see that the parts of the DNA, that the locations where the DNA is, are not the locations where ribosomes are. You know, well, maybe with this light is not very clear, but you know the signal of the ribosomes do not overlap with the signals with the DNA. Okay, so that means that the ribosomes at the DNA are separated physically. Okay, and remember that the DNA and the RNA polymerase are together instead. Okay, so that tell us that there is a kind of separation between the different stages of the gene expression flow. You have first to have transcription, and then you have to have translation, and they happen in different locations of the cell, and that's something that is quite important. So because, well, in this case, for instance, you see that the location of the RNA polymerase, of the ribosome, excuse me, is different, entirely different, and you know, excludes completely the locations of where the ribosomes are. DNA and ribosomes don't mix intracellularly. Okay, so in fact, when you label simultaneously RNA polymerase and ribosomes, then you see that they occupy different niches, different locations inside the cells, what is quite interesting. Okay, so where are the transcripts? Well, to monitor, to visualize where the transcripts are, you can also use technologies. For instance, you can use fluorescently label a single-stranded DNA that under given conditions hybridizes where the mRNA is. So for instance, in this case, this is a long transcript, and we had to use 48 different small oligos that could hybridize enough length of mRNA to visualize where the molecules were. And again, what you observe is that the RNA is not all over the place. It is located in specific places of the cell. So all these images, what are telling us is that you know, not everything is everywhere. Inside the cell, you have a structure that separates different components of the gene expression flow. You can see that in the chromosome, in a plasmid, but also in the chromosome. I mean, the technology now enables you to take a specific transcript of your choice that is here, and then by making this trick, you can distinguish where things are. And if you use oligos with different color codes, you can even see what is what and where things are in a cell. Okay, but the bottom line is not everything is everywhere. There is a considerable granularity inside the cells, and if you then combine different colors and different primers and different marker proteins, then you can really visualize where different things are in a cell. Where is the plasmid? Where is the transcript? Where is the RNA? Where are the ribosomes? So this indicates, not only this bacterium, but in many other bacteria, that the structure of the cells inside is not homogeneous, just on the contrary, is highly not compartmentalized, because in bacteria you don't have compartments, but it's highly domainalized. So you have various domains that do not mix with each other. Okay, well, you may say, well, this is nice, so that's the end of the story. Well, it's not quite the end of the story. I don't know how many of you work with microscopes, and well, we are, absolutely fascinated by images, and one image is better than 1,000 words, but you have to be cautious, okay? Sometimes images can be very, very misleading. So, for instance, in this case, you may think, what do you think you have? You think that you have an spiral here? Yes, right? It looks like, well, it's not an spiral. So they are concentric circles, and it's just an optical, say, illusion, right? Because our brain is unable to decode what we see in terms of concentric circles, and we see a spiral. Here, the same, okay? So are these two, planes at the same level? Okay, well, I don't know. I really don't know. I mean, it's something that you can create. It's an illusion that you can create in a drawing, but not that. But things can be very, very much worse, okay? And images can be misleading to the point that you can confound a muffin with a chihuahua, I think that can be really bad, okay? So I think that at some point we have to use other methods to examine what happens inside the cells that do not rely on images, that rely in other things. And one of the methods that are being used, and we have used it, and others have used it, is to try to get information from the phenomenon called noise. What is noise? What is gene expression noise? Well, gene expression noise is the phenomenon that is connected to the fact that at any given time, not all cells are expressing the same gene at the same level, okay? So you have a gene and you have a population. And then you can make an average of the population, but if you go cell by cell, what you find is that not all cells are doing the same level of expression at the same time. So that happening cannot be seen just by looking at a culture. You have to go one by one. For instance, in this case, you have what is called a monomodal distribution of gene expression, and then what happens is that every cell at a given point expresses the same gene, but at different levels here and there, okay? And therefore you have an average, and if you measure the expression of the population, then what you obtain is this average here, okay? And we'll kind of symmetrical distribution of the thing, but visually you will just get this growth curve or this induction curve, and then the expression would be this one, despite the fact that at the population level you have this noise and this dispersion in the levels of gene expression that are invisible to the eye unless you monitor one by one what is going on. But what may happen also is that you may observe the same level of expression, but if you look at the level of single cells, instead of having these kind of modest variations at both sides of an average, you may have tremendous variations to the point that some cells can be expressing the gene at very high levels, and others may not be expressing the gene at all. And even though macroscopically you detect the same curve here and the same curve here, at the level of single cells, what you have is this bimodal distribution of gene expression in the two circumstances. What do we argue? What is the connection between that and the intracellular structure that I just mentioned? What we argue is that these signals, this noise, in reality is telling us something about what is going on inside the cells. So it's like the cells, by having this diversification in their intracellular, in their gene expression, is telling us something that happens inside the cells. What is what happens? This is our hypothesis, and we argue that this is a good one. So what happens is that you have a medium in the cell that is quite granular. You have many obstacles, and you have many things that are, like molecules and complexes and everything. Then for a promoter to work, it needs a transcription factor. But the transcription factor might be produced from somewhere else in the cell and has to find the promoter for activity. So what we argue is that it's likely that the noise that we observe in these gene expression patterns reflects in reality the itinerary of the transcription factor until it finds its destination in the promoter. So in other words, what we see as a noise, in fact, reflects the physical qualities of the container, the physical, the physical surroundings of where the promoter and the transitional factor are, in such a way that if we were able to decode this noise, we could know something about the shape and the physical characteristics of the location in the cell where this gene expression event is happening, okay? So that's the bottom line of that. So instead of looking at the cells, we focus in an indirect signal that we argue is connected to the physical structure of the cells. Why the physical container is super important? Well, because it happens that the physical container determines predictability. So if you have a billiard table, and I don't know, some of you like this game, you can make an absolute prediction of what would be the consequences of having this ball being like pushed into this one. You can make a perfect mathematical prediction of why, of how this ball will hit this one, this side, and then it will hit this other. And you can make that prediction because the physical receptacle of where this happens is formed of straight lines, and therefore you have a highly deterministic scenario to predict what happens. But if you use a different table, then things become more difficult, because you have now, you enter into a chaotic situation. So chaotic situation means that very, very, very small differences in the parameters of the hit of the ball in one of the sides will have later tremendous consequences. And therefore, you hit this ball into this one and to the other, and ladies and gentlemen, no one can predict what's going to happen, right? So by the same token, in a cell, depending on the shape and the characteristics of the physical, say, setting, then gene expression can be very, very predictable or can be completely unpredictable, okay? So because of that, understanding what is going on inside the cell in terms of the intracellular architecture is so, so important. So what happens when you look at promoters? Well, what happens is that some promoters are monomodal, so that means that when they are, I just take two, these two promoters are inducible by benzoate in Pseudomonas putida. So when you induce them, what you see is that the entire population, like obediently goes and moves in one direction. So these are promoters that are called monomodal, because basically the fluctuations of the diversity, the noise that you obtain in these cases is little, and then they are just little variations in respect to an average that is more or less sharp here, okay? But in other cases, some other promoters are bimodal. So for instance, in this case, another promoter of Pseudomonas putida, I will not go into detail, instead of having the whole population going like swiftly from one place to the other, what you have is that the population, when the promoter is induced, jumps directly from this state to the other state, nothing in between, okay? So that means that at any given time, part of the population is off and part of the population is on. And the same happens with other promoters. So if you remember the story about the toggle switch, this is a natural toggle switch, and obviously the mechanism that is behind is completely different of the one that was engineered in the laboratory of Jim Collins that ago, but this is a toggle switch, because part of the cells at some time are on, part of the cells are off. And the induction, instead of going little by little, involves a direct jump from being off to being on, nothing in between, as I said. Okay, so you can visualize that very nicely, not only with cytometry, but also directly by putting a promoter, by putting different reporter systems in front of these promoters. And for instance, in this case, we have a situation here. We have a promoter that have studied a lot that comes from P. putida. And in this system, we have two promoters in the same cells. And depending on the circumstances, then one promoter is on, the other is off, and you can see that at a given moment, then this creates a phenotypic heterogeneity, okay? Because that means that at any given time, not all cells are doing the same some cells are doing metabolically some things, and others are doing metabolically a different thing, okay? So that's something that is quite remarkable, and is also important for metabolic engineering and other types of engineering, because what you have in a population that you believe is clonal, and that every cell is doing the same thing, turns out not to be true. What turns out is that you have a division of labor, because at sometimes, for instance, if they are metabolic promoters, it may happen that part of the cells are running a part of the metabolism, and other cells are running another part of the metabolism, and this is something that you cannot detect by other means, because they are all clonal. They are all genetically identical, okay? So this is one of the tricks that bacteria use all the time to improve their metabolic capabilities, that instead of having everyone doing everything at the same time, you have a division of labor. So having this heterogeneity is a trick, an evolutionary trick that populations have to improve their capacity to deal with different nutrients and with different conditions. Also, the capacity to deal with new conditions, with new environmental conditions. This is what is called, in some cases, bed hedging. So, because you have a phenotypic heterogeneity, if the selective pressure that you have on this population is resisted better by one strain or by one cell than by the other, this strain may survive and the others not. So this phenotypic heterogeneity can also be a mechanism of adaptation under different circumstances. Okay, so well, just to show you, visually, for instance, the fate of one cell over time, where you have these promoters, that one of the promoters that I showed you, that is bimodal. You go to one place or the other, one or the other, okay? So you start with a cell here, green, red, green some, red some, and at the end, well, the cells grow, and at the end you lose everything. But you know, in the way, instead of having, you start with a green cell, but then the green cell diversifies into red cells, green cells, red cells, green cells, and at the end, well, everything grows and so on. But you know, this is an indication that indeed the promoters are behaving differently. You can even, you have methods to monitor the fate of one single cell and see how it grows. And then, let me see how it is. Okay, you start with one cell. This is green, green, green. Well, a red one, green, red, green, red, green, red. And at the end, you have a mixture in the population of green and red, you know? So that means that because of this trick, where you have this heterogeneity intracellularly and this problem of this capacity to have a bimodal expression of some genes, then you have start with a completely homogeneous population, and then at the end diversify and having a phenotypically different population that all are descendant of a single cell. So that's something that is quite nice. Okay, so you say, well, what is the connection to the first part of the talk that I explained before? Can we leverage intracellular heterogeneity for optimizing genetic devices? Because all this story I told you about intracellular heterogeneity is based on this idea of optimizing genetic devices and use that as a tool for engineering things that work under the umbrella of synthetic biology. Okay, so I'll show you one case in which this is very clear. And in this particular case, what we wanted was to have an optogenetic control of biofilm formation. Unfortunately, I will not have the time to give you a lecture on biofilms. Biofilm engineering with the tools of synthetic biology is super exciting. I will be unable to talk about that, or maybe, I don't know if I have time, maybe the last day. But biofilms are no more and no less than bacterial communities that stick to surfaces, okay? And when they are in surfaces, it's very interesting. Well, sometimes it's painful, and sometimes it's interesting. It's painful because they are, in many cases, pathogenic bacteria stick to surfaces, and that's a problem. But from a biotechnological point of view, having biofilms is interesting because you can create catalysts that are associated to a solid matrix, okay? So playing with engineering biofilms is one of the kind of least to do things in in synthetic biology. Well, in any case. So it has been known for many years that the reason why many bacteria form biofilm or not is connected to the intracellular levels of a molecule that is called cyclic DGMP. So this is an intracellular, signal that goes up when bacteria go into a biofilm state and go down when bacteria go into a lower state. So how can we engineer bacteria, Pseudomonas of course in our case, to stick to surfaces when we illuminate them with light, and depart from that when we put them in the darkness? So look at the power of this thing, okay? So we engineer bacteria that can stick or unstick depending on whether we illuminate them or not, okay? So this is a phenomenal tool for a large number of environmental applications. So in this case, what we did was to take the same optogenetic device that I showed you before, but instead of having at the end GFP, what we had was another gene of interest that is a cyclase for GMP. So the idea is that if this cyclase is expressed, then you form a lot of the cyclic GMP, and then cells stick to the surfaces like crazy. If you don't have this enzyme here, then cells don't produce the cyclic GMP and they don't form biofilms. So we engineered this in front of all these bunch of genes that were the same that we used before for GFP, and again we have a large number of parts that you have to optimize. 1 Schendel-Garno, 2, 3, 4, 5, 4 promoters, and a bunch of terminators. Okay, again, a typical optimization problem, with the addition that we had to not only to play with transcription, but also with availability of substrates for this enzyme there in the cell, okay? So similarly to what we did before, one can think on optimizing the pathway by diversifying the intergenic regions, and this is what we made before. And well, if you do that, you can get indeed in a plasmid, for instance, some variants of this construct that will form biofilm where cells are illuminated. This is, for instance, a very nice experiment. This is a plasmid, and the plasmid can be diversified with MAGE, as I showed you before, and then it can be optimized, and then you see that under these circumstances you form a thick biofilm when you expose the cultures to light, and then you don't form biofilm or very little biofilm when cells are kept in darkness. But as I said, we want to have a further round of optimization connected to intracellular granularity. So the basis of that is the following. In different locations of the cell, well, you have a chromosome, but not only chromosome. You have, I mean, physically, you have the distribution of the chromosome, but the chromosome, because of this intracellular granularity, will not see at any given time the same resources for the gene expression flow. In any given position, it will happen that at this position, you have a given level of DNA supercoiling that influences transcription, you have a given level of RNA polymerase, a given level of ribosomes, a given level of metabolites, a given level of transcription factors. So that means that depending on where you are in the cell, where your genes are in the cell, the device will be exposed to different resources for its expression, okay? It's like, you know, if you want to kind of explore a new continent, then not all places are good places to harbor, you know? Some places are very bad for harboring, and other places are optimal for harboring, you know? This is one of the Columbus images, looking for a good port to establish their first colony in South America, okay? So by the same token, what we can do is to take the device and shoot the device into the chromosome of your bacterium, and then the expectation is that depending on where the device goes in the chromosome, it will find different resources and different, say, contexts that may further improve the behavior of the system that you want to optimize, okay? So what we did was to use one of the mini transposons that I discussed in previous days. We engineered the device in one of these mini transposons, and then we shooted the device in different chromosomal locations in an entirely random fashion. So we generated large libraries of insertions, and remember that the phenotype that we are after is a phenotype that consists of formation of biofilm in the presence of light when cells are illuminated with light, and no biofilm when they are kept in the darkness. So this is what we obtained in one of the experiments. So you see that most of the insertions didn't work at all, and the explanation for that is that once you move from a plasmid into a transposon, the whole landscape of parameters change, and therefore you have to look for another set of parameters that in this case is delivered or is provided by the context that the insertions find in their way to the chromosome. So for instance, in this case, we have some of them that are very good. I would say that this one, for instance, is very good. You have a 300-fold increase in their formation of biofilms from darkness to illumination, and others do a bit worse. Okay, but in any case, this is a kind of good, say, indication on how having the device being inserted in different chromosomal locations is also a tool for optimization in the cases where we cannot really control all the parameters. So this is a similar experiment. The difference is that in this case, the host strain was mutated to avoid entirely formation of biofilms under natural conditions. So it carries a mutation that is called WSP, that is connected to a system for response to contact and so on. So that's deleted, and therefore the only way that bacteria have to form biofilms in this particular strain is if you artificially increase the intracellular levels of cyclic DGMP, and therefore you bring about more adhesion of the cells to the corresponding surface. Okay? If now you look where the different insertions are, you observe with no surprise that many of all the insertions go to different locations of the chromosome, and you can take a look to what specific sites there are already. So you have an entire variation of different localizations. One added value or one added bonus of this approach is that the insertion itself may hit a gene that might interfere with the expression of the phenotype. And if you hit that gene, then the phenotype gets expressed at a much better level. So that's something that indicates that you have all these levels of possible diversification, of all this exploration of a solution space that goes from mutating everything to mutate a small part, to change the chromosomal location, but also to change or to alter with this procedure the genetic context. And it is not impossible that some of the insertions that you have here have hit a gene that otherwise would inhibit formation of biofilms. Okay? Okay, so I think that was the story for today. I think that, to be frank, today has been one of my favorite classes. This is one of my favorite stories that I like to share with the audience. And well, I hope that I convince you that you can play as a genetic engineer or as a synthetic biologist equally happily with direct design and with evolution, and there's no conflict to use the two approaches for getting your right construct. Okay? So that's all for now. And then we'll go for a coffee break, okay? And we'll be back in half an hour, maybe a few minutes after 4:30, okay? Well, we'll be here anyway. Okay, thank you.